In a random spontaneous process known as depurination, the nitrogenous bases known as purines, adenine and guanine, can be removed from the DNA backbone and are often replaced at random, which then may lead to an error and thus a mutation. X-rays can break the sugar-phosphate backbone of a DNA molecule, and this can cause a chromosomal break and thus a chromosomal mutation. Ultraviolet light can cause two thymine nucleotides, which are adjacent to each other, to adhere, thus forming a thymine dimer. The next time the cell replicates, these two bases may be interpreted as being a single nucleotide, and thus this would cause a mutation. When a transposon inserts into a new area of the genome, it can disrupt the DNA which is there. So it may disrupt a gene, it may separate a gene from the elements which control its expression, so the movement of transposons can result in mutation. When retrotransposons are inserted into the DNA, this can interrupt a specific gene, or it could separate a gene from its control elements, and thus this can also be a source of mutation. When the sequence of an endogenous virus is inserted into the host chromosome, this can disrupt the DNA region where the insertion occurs, and thus cause a mutation. Heat can cause deanimation, the loss of an amino group, and this could convert a cytosine nucleotide to that of uracil. Uracil would bind to adenine rather than guanine, and this would then cause a mutation. There are a number of reactive oxygen molecules known as reactive oxygen species. This can be formed during inflammation, can be the result of air pollution, of exposure to cigarette smoke. There are a number of causes of reactive oxygen species. They can react with nucleotides and change their base pairing.